So the female gametophyte. Female gametophyte. So female gametophyte is nothing but the gynoecium. So already we have seen the male, which is the androecium, and the gynoecium is the female gametophyte. Okay, so this is the female reproductive part of a flower. So now here we have an important term which is called as what? Pistil. Okay, so like the carpels you can say. So generally either this carpel may be present as one in number or many. Okay, so depending upon that, for example, if this is a if this is consisting of only one pistil or carpel, then it is termed as monocarpellary. And if it is having more than one, then it will be called as what? Multicarpellary. Okay, so if it's in multicarpellary form, then what will happen is the pistils, either they may be present how? In a free form or in a fused form. So if it's a free form, okay, then we call it as what? Apocarpus. Apocarpus. Whereas if it is in the fused form, we say that is a syncarpus pistil. Okay, so each pistil is going to have certain parts where the female gametophyte or female gamete, this egg, which is going to get formed and the fertilization is going to take place. Okay, so let's see what are the parts of a pistil. Parts of pistil. Okay, so generally pistil consists of 3 parts. Stigma, style, ovary. Okay, these are the 3 important parts of a pistil. So now I'll just, yeah. So if you see, I'm just roughly drawing this. This is the female gametophyte. So this is the stigma. This is the style. Then here we have the ovary. And this is the thalamus. Okay, so sigma, style, ovary. So the first part, if you see, it's the stigma. So stigma, what it generally does is, it receives the pollen grains. What is the function of stigma? It is going to receive the male gametophyte, that is the pollen grains. Second one is the style, as you can see over here. It is an elongated, slender part which is present below the stigma. So through which what happens is, it just acts as a stalk, like the stalk of the flower. This also gives a connection between the stigma and the ovary. Then the last one is the ovary. Okay, so ovary is nothing but the at the bottom or at the base part, you will see this bulged part. Okay, so it's the bulged basal part, which is going to contain an important thing called as a placenta. Containing placenta. Okay, so this particular placenta is located inside the cavity of the ovule. Located inside the cavity of ovule. Okay, so these are what certain parts or what a carpel actually consists of. Okay, now this placenta actually is going to consist what? The megasporangia or the ovules or the seeds, you can say. Okay, so this placenta only, it consists of what? The megasporangia or ovules, which is nothing but further which is going to develop into what? The seeds. Okay, so now how many number of ovules will be present? It may vary from plant to plant. Okay, like for example, if it is suppose if only one ovule is present. So the plants which you can take over here is what? Mango, wheat, all those plants. Okay, they will be having only one ovule. If you take a mango and open it, how many seeds you will find? Just one big seed, right? So that's called as what? One ovule will be present. But if it's more than one ovule, examples, what you can take? Many are there. You can take papaya, or either you can take the flower. In flowers, if you can take the orchids, again watermelon, and so on. So okay, so depending upon this, whatever how many ovules are present, it will vary according to the species, whether one ovule may be present or many may be present. Okay, so in order for this ovule or the megasporangia to form, okay, let's see what is the procedure, how the megasporangia is formed. Megasporangia. Okay, so if you see the structure of a megasporangium, okay, generally, I'll just write over here. So generally here you will be having a small embryo sac, and then rest of the things attached like this. Okay, so this central part I'll be elongating afterwards. Okay, I'm just drawing it roughly just to show you what is a megaspore. Okay, so here it comes like this. Megasporangia. Okay, so let's label this now. So this particular thing is the embryo sac. There will be two ends over here. So this is the chalazal end. Okay, and this one will be the micropylar end. Okay, then you will be having two teguments. These are the two teguments which are outer and inner integuments. Okay, then this region over here is called as the micropyle. And then here, this is the funicle. And here inside is the hilum. Okay, now let's see what are these parts one by one. So now if you see over here, we are talking about the seed, that is the female gamete. Okay, so here, if I take the first one, now whatever ovule is there, okay, they are generally attached to this placenta. I told you inside the placenta only this particular things are present, whatever is drawn over here. So what happens is, whatever the ovule is there, that will be attached to whom? The placenta. Right? So whatever the attachment is there, that takes place by this structure, which is called as the funicle. Okay, so placenta by what? Funicle. Now, obviously, here you can see there's a junction between the funicle as well as what? The ovule. Okay, so whatever the junction of the ovule and the funicle is there, that particular thing is called as what? Hilum. Okay, so junction of ovule plus funicle is called as what? Hilum. Okay, then I told you two integuments or the protective coverings are present over here. So each ovule will be having what? Each ovule has one or two protective layers or envelopes. Protective layers or envelopes. So these are only called as what? The integuments. Okay, that is outer and inner integuments. Okay, so here everything will be covered, okay, except one part which you can see over here, the small part which is there, that is a micropylar. Okay, so whatever the protective layer is there, the outer and integuments, this particular things will cover the entire thing except the small opening which is present over here, which is called as what? Micropyle. Okay, so covers the ovule, leaving the small opening, that is the micropyle. Then as I told you, two ends. So near to the micropylar end, opposite to that over here, if you see, it's called as a chaliceal end. Okay, so chaliceal end means it will be down. That is the basal part, you will find it. Then you have the next thing, which is called as what? Nucellus. Okay, so like if you see here over here, this part, this empty, this entire part, this is only called as what? Nucellus. Okay, so where is nucellus present? Obviously, it's present in within this integuments, and it's actually it contains a mass of cells so that it can store what? Food materials. Okay, so it stores what? Reserve food materials. It reserves food materials. Okay, so together we have this entire thing which is called as what? The embryo sac. Means whatever the female gametophyte is there, that is only called as what? The embryo sac. That is here if you see this thing. Okay, this is what? The embryo sac. That is the female gametophyte which you will find. It is located inside the nucellus because nucellus is the place from where it's going to get the nutrition. Okay, now let's see how actually this megaspore, okay, is formed. So obviously the formation of the megaspore, like we saw in the microspore, the formation of the microspore is called as a microsporogenesis, which is formed from the microspore mother cell. In the same way, over here, the only difference is instead of the microspore, you have to write the megaspore. Okay, so let's see the process of what? Megasporogenesis. So genesis means, already I told you, it means the formation. So over here, what happens is megasporogenesis is nothing but the process of formation of what? Process of formation of megaspores. From whom? From the MMC, that is the megaspore mother cells. Okay, megaspore mother cells. So generally, whatever the formation of the megaspore or the megaspore mother cell which is there, it will be large because obviously it's a mother cell. So that MMC will be what? It will be large, plus it will be having a dense cytoplasm. Correct? With a prominent nucleus. Prominent nucleus. Right? So over here, generally, whatever the process or the differentiation of this megaspore mother cell is there, that is by the meiosis process, it generally takes place in which type of region? Now, there are two regions, micropylar and chaliceal region. So, whatever the MMC differentiation is there, that is the divisions which it is going to undergo, will be taking place in which end? Micropylar end. Okay, so whatever the ovules or the seeds which are going to be formed, they are going to differentiate where? In the micropylar region of the nucellus. So gradually, whatever the MMC is there, it will undergo the meiosis process. The meiosis process. And when it undergoes the meiosis process, it is going to produce 4 megaspores. Okay, so out of this 4 megaspores, 1 megaspore is going to be functional. Means it is only going to lead the entire process, whereas the other 3, they will degenerate. Fine. So ultimately, how many megaspore out of 4 you will be having, which is functional? Just 1. So we are going to see further whatever the process is there only with regards to the 1 functional megaspore, right? So let's see the thing. So now here, as I told you, single functional megaspore will be there. Single functional megaspore. Now this particular single functional megaspore is only going to develop into what? The female gametophyte. Right? So only here single like megaspore is going to develop into a female gametophyte. So this kind of development is termed as monosporic development. Monosporic. Important one. Monosporic development. Okay. So now, as I told you, it contains nucleus, right? So whatever the nucleus is there, it is going to divide. Okay. So the nucleus, what will happen to the nucleus of the functional megaspore? It is going to divide. How? Mitotically. So when it divides mitotically, it is going to form 2 nuclei. Okay. So what happens to this 2 nuclei? This 2 nuclei is going to go to the opposite ends. Now already we know 2 opposite ends are there. So these are going to move towards where? The opposite ends. Right? So now over here, whatever the structure you're going to get is a 2 nucleate embryo sac. Now this particular 2 nucleate embryo sac is going to undergo 2 more mitotic division. So 2 more means from 2 it will become 4 nucleate, and from there 1 more, that is 8 nucleate embryo sac. Okay, so I'll just draw the thing earlier, whatever the small thing I drew there inside the nucleus, that I'm just going to enlarge over here. So here, okay, now this is your, consider this as your embryo sac. So now here, this is the antipodal. This is the central polar nuclei. Okay, means this is the polar nuclei, that is the central cell, and this is the egg. Okay, these two things which you can see is the synergids, and then this is the finger-like apparatus called as filiform apparatus. Okay, so from the 1 nucleated state, it will undergo a mitotic divisions where 2 nucleus, then 4 nucleus, then 8. So, okay, so 8 nuclear embryo sac we are going to have finally, right? So now what happens is after the 8 nucleate, and this is the structure actually how we can see the thing. Okay, if you enlarge it. So what happens is like immediately after the first nuclear division, what happens? There the cell wall is not going to form. When the cell wall will form is after the 8 nucleate stage, that is at the last stage, just now which we saw, the 8 nucleate stage, then only whatever the cell walls is there, that will be laid down. Okay, so this is the important point over here. That is, whatever the 8 nucleate stages there, here the cell walls are laid down. Are laid down and the embryo sac gets organized. Gets organized. Right? So now what happens is we have the 8 nucleate members, out of which whatever the 6 of the 8 nuclei which is there, it is going to get surrounded by cell wall. And whatever the 2 we have, okay, the remaining 2, because only 6 I told you. So remaining from 6, 2 are there. So whatever remaining 2, which is called as what? Apolar nuclei, that will be present below the egg apparatus, below egg apparatus in the central cell. Central cell. Okay, so 6 nuclei which we have, or the 6 cells which are there, out of those, 3 of the 6 cells, they are placed at one end, which is called as placed at micropylar end. Okay, so this particular 3 of the 6, see 8 we have, okay, so out of which whatever the 6 is there, that is surrounded by the cell wall. Okay, so you will be having 3 at one end and another 3 at the basal end, that is the base end. And then in between you have a central cell which consists the rest of the 2 polar nuclei out of the 8 nucleus stage. So now whatever the 3 cells which you are going to have out of the 6 cells, 3 will be placed at the one end, which is the micropylar end. Okay, so that particular micropylar end is going to have what? The egg apparatus. Okay, together it's called as what? The egg apparatus. Okay, so it will be having 2 synergids plus 1 egg cell. This is very important. Okay, 2 synergids plus 1 egg cell. Right, so this synergids, if you see over here in this is the synergids, 2 synergids you can see over here, right, at the micropylar end. So whatever the synergids are there, they are going to have a thick coverings, okay, at the micropylar end, and whatever the they are fused together by what? The apparatus, which is called as the filiform apparatus. Okay, so this particular synergis is going to, like, what do you say? It will help in leading the pollen tubes into the synergis. Whatever this filiform apparatus is there, its function is what? Whatever the pollen grains is going to come, okay, it will lead it inside into what? The synergis. So that is the function of this filiform apparatus. Okay, so 3 cells we saw, which is at the micropylar end. Now remaining 3 is there, which will be present where? The opposite end. Remaining 3, that is the chalazal end. So these are called as what? Antipodal cells. So if you see here, this is the antipodal cells. So this will be the chalazal, this will be the micropylar. Okay. So now, yeah, antipodal cells. So typically, the last thing which you're going to get, okay, that is a typical female gametophyte. If you see a typical female gametophyte. So typical female gametophyte is how? 7 cell and 8 nucleated. Now don't get confused that there are 8 nucleus, so how only 7 celled it's called as. Okay, see, I'll show you. Now, if you see over here, okay, now here how many nucleus we have? Just a second, just change the pen color. Yeah. So here you having 1, 2, 3. Okay, here 2. So 4, 5. Okay. Then here 6. Okay, so how many cells together? 1, 2, 3, 4, 5, 6, and 7. So here how many cells are there? 7 cells. Okay, now this is considered as one cell, this particular thing which is there, one cell it is considered as. But here the nucleus will be 8 because 1, 2, 3, here 2 will be there. So 4, 5, 6, 7, 8. So hence it's called as what? 7-celled, 8-nucleated female gametophyte. Okay, so now I told you that the female gametophyte has to reach to the male gametophyte has to reach to the female gametophyte in order to continue the process of the fertilization. So for this, one important process is required, which is called as what? Pollination. Pollination. So simple thing, pollination, we all know it is nothing but the process of transfer of pollen grains from anther to stigma. Transfer of pollen grains from anther to stigma. So anther, you know, it's a male part, whereas stigma is a part of the female gametophyte, right? So depending upon what type of pollens they are, okay, means what the source, how it is going to get transferred. There are certain pollination types as well as the agents which help in what? The pollination. Okay, so first we are going to see about the source of the whatever the pollens are there, that one. Okay, so it all depending upon the pollen source. Okay, it has been divided into main 3 categories: autogamy, nitogamy, nogamy, and xenogamy. Okay, so now what is this autogamy, nogamy, and xenogamy? So autogamy means, now pollination means in general, we already know transfer of pollen grains from anther to stigma. But what are these different terms? Okay, so autogamy means here the pollen grains are transferred from anther to stigma of same flower. Same flower. Okay, so because of this, whatever the flowers are there, okay, it should be in that whatever the anther is there, it should be very close to the stigma. Anther close to stigma. Okay, so hence they are called as what? The autogamy process. Nitinogamy, in this what happens is, here also the transfer of pollen grains from anther to stigma only, but anther of one flower to stigma of another flower, but same plant, in same plant. See, now in the autogamy, pollinating agents is not required, like water, wind, birds, or something. Whatever the agents are there, they're not required because they're in self only, in one flower only, the pollination is going to take place. But in the nitinogamy, what happens is, in the same flower, means in the same plant, 2 flowers will be present. So, one anther, then to the stigma of the another flower, but in the same plant. That is the difference. Whereas the last one, which is the xenogamy, here it's like the nitinogamy only, but instead of the same plants, you have to write the different plants. Okay, so anther of one flower to stigma of flower of different plant, of a different plant. So for this, the agents like wind, water, whatever is there, it's very necessary. Okay, so this is all depending upon the dividation of the pollination. So how the pollination or how the pollen grains are going to get transferred, whether it's to the inside the same flower or within the same flowers of same plant or different plants. Okay, so depending upon that only, these three types are there.